very excited about reprogramming pluripotent stem cells. We have three main interests in the lab. One is mechanisms, as Evan alluded to, and decided to tell you more about that. But we also are very interested in developing human cell models to study diseases, in particular neuropsychiatric diseases. But we also actually have an Alzheimer's project. And also would like to think into the direction of actual regenerative medicine and using reprogrammed cells for a direct therapeutic implication. So as I said, we are generally interested in the lab in the molecular mechanisms that define cell identity, and in particular the formation of neurons. When you think about it, it's really the chromatin configuration and a specific set of transcription factors that are being expressed in each cell that we think are the molecular substrate of cell lineage identity. Just put this slide up here. Again, as Californians, you know this gentleman. Just to remind everybody that the epigenetic information is really, really important. You can have the exact same set of genes and look very different. So the same should be true for cells. So when you think about when a neuron is formed, there are really three cell biological steps that these cells typically have to undergo. First, somehow the lineage has to be induced. Then typically cells have to mature. And once the neurons are made, there must also be mechanisms in place that guarantee the stability of the acquired cell identity. So let me start with the question of how are neurons actually induced. And following the famous dictum by Richard Feynman, what I cannot create, I do not understand, we set out to try to generate neurons from scratch. And we tried to convert completely unrelated dermal fibroblasts to convert directly into neurons, and this experiment worked. We found these three transcription factors, ASA1 with one like NBRIN2, that quite efficiently induced these, we call them IN cells or induced neuronal cells. And the remarkable observation was that these cells not only looked nice and had some neuronal genes expressed, they actually had the two main functional properties of neurons, namely were able to fire action potentials and form synaptic connections. The functional characterization was performed in close collaboration with Tom Sudov's lab, and we are collaborating ever since. So over the years, we have explored several donor cells and found that very similar sets of transcription factors actually can convert a variety of different cell types into these ion cells. And I guess I'm most proud to be able to tell you that we also managed to convert adult human lymphocytes into these neuronal cells. This was accomplished with the combination of these four transcription factors, the three I mentioned in addition to NGN2. We also found a set of small molecules that further improve the reprogramming process. And of course, we always make sure that these cells are functional and they can integrate into pre-existing neural networks and form synaptic connections with them. So let me now focus on more the mechanistic sides, now that I've hopefully demonstrated you that it is actually possible to induce the neuronal lineage from various donor cell types. But how does it work? It's just a small handful of transcription factors that apparently seem to be at the very top of a hierarchy of a lot of cell biological processes that have to happen when you convert a flat, boring-looking fibroblast to this elegant cell of a functional neuron. So let me start with this factor called ASA1, which is a BHLH, a basic helix loop helix transcription factor. Here you see the crystal structure modeled after MyoD, a related BHLH factor. And it typically forms a heterodimer with constitutively expressed A proteins. And as you can see, the two helixes are primarily there to interface between the two partnering transcription factors, and it's really the extension of one of these alpha helixes that creates the basic domain, which is binding the DNA in the major groove. So one of the first things we asked, where is ACE1 actually landing in the fibroblast genome? So we just took mouse emery fibroblasts, or MEFs, infected them with the 3 transcription factors, and then pulled out ACE1 with antibodies and did a ChIP-seq experiment to map where the binding sites are. And we got a nice list of about 6,000 or so sites that are significantly enriched. And the remarkable observation was that when we compared these sites to where ACE1 is physiologically bound in neural precursor cells, and those are endogenous ACE1 levels, we see a very similar binding pattern. So that was completely unexpected, because your fibroblasts are considered to be in a very different epigenetic state, and you would expect that any transcription factor ectopically expressed, like a neuronal transcription factor, should be completely lost there, right, and not be able to find its proper neuronal targets. Rather, sort of, try to deal with the situation and find, some open, accessible sites in the chromatin of these fibroblasts. So that was really surprising, why ACL1 would bind very similarly. We first made sure that this is actually a property of ACL1 itself. It doesn't require the other 2 factors, so we infected the fibroblasts with just ACL1 alone, and again, see the very same binding pattern. So somehow ACL1 is able to do that. We're wondering, well, there's really only 2 possibilities how this can happen. Either these fibroblasts are somewhat weird, and all these neuronal targets happen to be accessible, and the chromatin state is compatible with binding of a neural transcription factor. The alternative hypothesis is that the chromatin is actually different than in NPCs, and these neuronal targets are closed, but ACE1 has sort of special properties, properties of what we call a pioneer factor, which is the property to access closed chromatin. And to distinguish between these 2 possibilities, we just looked at the chromatin configuration at the ACE1 target sites in completely unperturbed fibroblasts. So we knew, of course, where ACE1 will bind, and took those sites in the genome and asked what's the configuration of these sites in the fibroblast before ACE1 is showing up. And what you see here in this, on the left, is a FAIR-seq analysis, which is a way to measure nucleosome-free regions. We see a clear depletion of this signal here at the ACE1 target sites, suggesting that the vast majority of these ACE1 targets are actually in a closed configuration. And we come to the same conclusion when we pull out nucleosomal DNA associated with histones by pulling out histones with H3 antibodies, and now we see a clear enrichment of the signal. So it's very clear. The fibroblasts are not weird. They are actually different in their epigenetic state, and the ACE1 target genes or sites are actually closed, and ACE1 is able to bind these sites regardless. And in contrast to many pioneer factors that have been described, I just showed you earlier, ACE1 is actually able to not only bind random closed targets, it's actually proper physiological binding sites. That's why we coined this term that ACE1 must be a so-called on-target pioneer factor. All right, so what about the other 2 transcription factors? We also looked at BRAIN2 and its binding pattern in fibroblasts, either alone or in combination with the other 2 transcription factors, or where BRAIN2 is bound in neural precursor cells. And as you can see, the binding pattern is completely dependent on the circumstances, on the conditions. When we do like a Venn diagram overlap, there's hardly any overlap between these the BRAIN2 binding sites in these 3 different conditions. So very unlike ACL1, BRAIN2 is highly context dependent. It actually turns out that the binding sites in MEFs, when you co-express ACL1, actually enrich for an ACL1 motif. And there is quite a number, about a quarter or so, of the sites that are co-occurring now with the ACL1 binding sites. So these sites don't even have a BRAIN2 motif, but still BRAIN2 is going there, suggesting that ACL1 is dragging somehow this other reprogramming factor to these proper sites. And not surprisingly now, when we look at the BRAIN2 binding sites in either of these conditions, they tend to be open and accessible. So we arrive now at this model here that the AS1 target sites must be in a closed chromatin configuration, and somehow, we have of course no idea yet, but somehow AS1 finds its targets and binds the DNA and probably recruits other factors such as BRIN2, but also assume chromatin remodeling complexes and so forth, and then opens up these sites to activate the neuronal program. So that is of course a nice diagram and taken from a review by Kenzeret. But can we actually watch this? Can we actually see that this is happening? And to do that, we resorted to this very elegant technique called ATAC-seq, which was co-developed by our close collaborator Howard Chang at Stanford. And that technique takes advantage of transposase to splice in sequencing primers into accessible chromatin. Because of this direct splicing of the sequencing primers, it's a very, very sensitive method. And as you can imagine, it's a very elegant way to distinguish accessible open chromatin from closed chromatin. So we first asked what happens to these AS1 target sites in the time course of a reprogramming. And we took several samples. In particular, early on, we looked at 12 hours, 24 hours, 36 and 48, and then later time points all the way to about 3 weeks after the conversion. And we separated the reads into long and short reads, because short reads are obviously associated with open chromatin, because transposase can access there, and it produces short reads. If the transposase bumps into nucleosome, it will only have a chance to splice in when the nucleosome, the other side of the nucleosome and the DNA wrapped around it, of course, is resistant to it. That's why longer reads in the ATAC-seq experiment produce the signal associated with nucleosomal or with closed chromatin. So we separated for this reason these two reads, and in black you see the short reads, so the nucleosome-free regions, and we see a very rapid induction of the signal here. So this is 12 hours after induction of mRNA of AS1, right? So we don't, the protein will be even later, and we can already clearly detect something is happening and opening up at these AS1 target sites. And this signal sort of increases over time. And when I can draw your attention to now the red signal, which is the reads associated with nucleosomal sites, maybe it's not so visible early on, but clearly by day 5, we see now these two bumps flanking the AS1 target size, the E-box motif here, suggesting there is now a stabilization of two nucleosomes flanking this open region. So I was really amazed by this data, because you literally can watch the conversion of the chromatin remodeling, essentially, from a more stochastically or a stochastic arrangement of nucleosomes to a highly structured, chromatin configuration after AS1 has been bound there. So this was just the AS1 target sites. What happens at the rest of the genome, and you can imagine with these genome-wide assays, a lot is happening. We were amazed to see that thousands of sites are opening up in the first hours. And as you could expect from the data I've showed you, the majority of the sites are actually opening up. And most of the AS1 target sites are found in this block here, but remarkably, there's also some sites that are closing down. And there's not even an AS1 molecule sitting there. So how this is regulated, we also have no idea. So let me now switch gears and tell you a little bit more about the third reprogramming factor, which is called MYD1-like. And unlike the other two, MYD1-like was really not well studied. When we found it to be a reprogramming factor, which is now a little while ago already, there were just 4 PubMed entries about MYD1-like. So what was known, though, was its beautiful expression pattern. This is just taken from one of these early papers, which demonstrated its expression pattern. And it turns out that MYD1-like is very tightly associated with neuronal identity. MYD1-like is induced very rapidly after neurons are formed, right essentially after the last cell division, when the neural precursor cells are dividing in the ventricular zone and the neurons are migrating up towards the cortical plate. All the way, so it stays as induced at that point and then stays on all the way through the life of the animal. There's two family members called MYD1. Sorry, the name is a little confusing. MYD1-like is the gene I'm talking about, and there's the name-giving family member called MYD1, which is also induced during the formation of neurons, but it's only transiently induced and then turned off again in neurons, and later induced in OPCs, in oligonucleotide precursor cells again. So for the OPC aficionados in the lab, in the room, sorry, it's a lab meeting, right? You will probably have heard of MYD1 already before, because it's a very prominent marker for OPCs later on. Hence, by the way, the name myelin transcription factor. So, all a little confusing. So again, we asked, where is MYD1-like going in these fibroblasts? There were no antibodies available, so we had to generate our own antibody, and luckily we're able to obtain pretty high quality antibodies. So we were able to actually interrogate primary neurons. In this case, it's a fetal brain that we used for this ChIP-seq experiment. Again, we got some nice peaks back, a motif. Remarkably, about half or so of the target sites are actually right at promoter regions, which facilitated our analysis quite a bit, because it was quite easy now to assign the target genes of the MYD1-like binding sites. And of course, we also did ChIP-seq in fibroblasts, where we expressed MYD1-like alone, which is shown here in the middle, or with the other two transcription factors. And as you can see, again, just like for ASA1, we see that MYD1-like is binding a very similar pattern. Well, I just tried to make the point earlier that this is something very unique and very special and not really much seen with transcription factors. So we thought, it's surprising, but maybe not so surprising that MYD1-like may also be such an amazing, pioneer factor like ASA1. It is a very powerful reprogramming factor after all, right? So maybe it's a very highly selected transcription factor. It's not a random transcription factor that we just picked, right? So we thought, well, yeah, it's probably also a pioneer factor. So we looked at the chromatin configuration just like before at these MYD1-like targets now in fibroblasts. And we're completely surprised to see that MYD1-like is not at all apparently a pioneer factor, because it turns out that unlike ASA1, which is shown in this red color here, the MYD1-like targets are actually completely open and accessible, because this is an MNase data set now here, so the lower signal or depletion of the signal clearly suggests that these sites are all open and accessible. So now we were wondering, how is that possible? You have this beautifully specific transcription factor. It's only there in neurons. It binds the same sites in neurons and in fibroblasts, but it's not a pioneer factor. So again, what's going on here? These sites are open and accessible. So it took quite a while until we managed to wrap our head around this. And the first sort of indication towards a solution came when we looked at the transcriptional output of MYD1-like. So we noticed that, in particular, early on in the reprogramming, we just expressed MYD1-like in these fibroblasts, that most of its target genes are actually repressed rather than activated. And that's also true and statistically significant when you take the entire list of MYD1-like target genes. So it's rather a transcriptional repressor, it seems. But I was kind of skeptical, because these RNA-seq data sets are genome-wide. They're it's 2 days after infection. There's a lot of things that can happen. Typically, there's cascades of transcription factors. So, is it really directly a transcription repressor? I had hard times to believe that, just because I thought, when you convert a fibroblast to a neuron, this is such a dramatic cell biological change. It cannot be sufficient to just relieve a repressor from the program, right? You may be able to de-repress things, but I would never thought that this is enough to really induce a new lineage. So I was really skeptical, and I challenged my German, I must say, postdoc, who was there at the time to do these experiments, Marius Wernig, to come up with a functional assay. We need, better evidence for this idea. So he took apart MID1 leg very systematically. Don't do all the structure function fun experiments, by the way, that led to other conclusions. But suffice it to say here that he identified the DNA binding domain, which are these two zinc fingers here right in the middle of MID1 leg, and turns out that this uncharacterized domain here is the most critical for reprogramming. But so he had identified the DNA binding domain, which is this small fragment, which alone doesn't really do anything. But he then fused this domain to a transcriptional repressor, ingredient repressor, or the VP64, a transcriptional activating domain. And then threw these constructs into our reprogramming assay, and here are the results. We measured two genes, TOG1 and tau, which are two neuronal markers. And all this data here are normalized to AECL1. So those are the efficiencies with just AECL1 alone. This is this little fragment here, which, as I said, doesn't really have much of effect. This is just the effect of AECL1 alone compared to the wild type addition of MID1 leg. And now, when we look at the transcriptional repressive fusion or the sort of the MID1 leg forced transcriptional repressor construct, we see, at least for the tau marker and to some degree also for the TOG1 marker, a clear improvement of the reprogramming over AECL1 alone, suggesting that repression actually helps. It's not quite as good as full-length MID1 leg, but it clearly supports. But now look at the activating version of MID1 leg. That completely kills the system. It's even worse than AECL1, the effects of AECL1 alone. So I had to change my opinion, and it's very clear. The main function, apparently, of MID1 leg is transcriptional repression. So now when you think about it, actually it does make, or it can make, actually a lot of sense. Because when you think, what has to change when you induce one lineage from an unrelated cell type? Of course, it's important to induce the target program, but somebody also has to take care of the original donor program. If you wouldn't do that, you would end up in some sort of dual hybrid cell identity, a sort of a fibroneuronal cell, which is probably not all that useful, right? So we thought, well, this is cool. AECL1 is this neuronal pioneer factor and use the neural program. Maybe what MID1 leg is doing, it represses the fibroblast identity. So we had various sort of approaches to address that question, and just pick, for the sake of time, just one of this, which is a gene set enrichment analysis, where we essentially overlapped the MID1 leg targets with a fibroblast signature gene, and as you can see, the sort of enrichment was highly significant, suggesting that this is exactly what happens, and that the main job of MID1 leg in the reprogramming process is to suppress the fibroblast program. So for the reprogramming function, we realized this is a beautiful solution that nature found for us. So we have AECL1, which is a great activator and very specifically and efficiently is able to activate the neural program. And then we have MID1 leg, which is clearly able to suppress the fibroblast program. And that's probably why these two factors work so well together in this reprogramming context. But we also asked, what could MID1 leg be doing in the brain? Isn't it a little unlikely that MID1 leg was just made by nature to suppress a fibroblast program? So we hypothesize, and that is shown here in this summary sort of overview, that MID1 leg is not only there to suppress a fibroblast program, but many other non-neuronal programs, including we have evidence, more specific evidence for a muscle repressor, but also hepatocyte and keratinocyte programs, perhaps even more. So it could be that MID1 leg is sort of the exact opposite of a sort of active neuronal identity factor, like ACE1, for example, would be. It also is a pro-neuronal factor, but it acts not by activating the neuronal program, but by silencing essentially all the remaining programs to allow factors like ACE1 to then activate the neuronal program. So a sort of inverted action to still have a net effect to being pro-neuronal. And yeah, so we really like this idea because it's a sort of a new type of function of a transcriptional repressor. Typically, repressors have been shown to repress specific lineages. I'm sure everybody of you have heard of the famous neuronal transcription repressor REST, which is there to essentially suppress specifically the neuronal program. And MYD1 is the exact opposite. Right? So it's repressing anything else except the neuronal program. So we thought if this is true, you would expect that the REST motif is enriched in neuronal programs, in neuronal promoters, and depleted in non-neuronal promoters, which of course is published, and we could confirm this as a positive control. But you would also expect that MYD1-like is the opposite. It's enriched in non-neuronal promoters and depleted in neuronal promoters. And that's exactly what we see. So I think quite strong evidence from just taking DNA sequences of the databases that confirms this or supports this idea. All right, but it also has, I think, important implications for normal development. Because one of the strong target genes that are repressed by MYD1-like is actually many members of the notch pathway, including the downstream effector genes HES1 and HES5. And that could solve a conundrum that has been out there since quite a while. I'm sure you're all familiar with the notch pathway and its function as lateral inhibition during normal neuronal development. And what the hypothesis is, or the textbook model says, is that you have a neuroprogenitor and a differentiating neuron sort of mixed in the ventricular zone. And the differentiating cells, they induce ACE1 at some point, which activates delta, which is the ligand for the notch receptor on the neighboring cell, which is still the undifferentiated cell. And notch would then activate transcriptionally the transcription factor HES1, which then in turn very effectively blocks ACE1. So that's a beautiful system to explain how ACE1 cannot be induced in these neural precursor cells, and explains how neural precursors and stem cells maintain their identity and keep proliferating. But it was not clear how these cells here can escape such a strong notch effect. After all, this is a strong sort of progenitor domain with probably lots of delta around. There's no reason to assume that just because these cells express delta, they couldn't also express the notch receptor. So why are these cells insensitive to the notch pathway and can still differentiate after all? And we think now with the MYD1-like link, this could sort of be the missing piece of this puzzle here, because somehow ACE1 is inducing MYD1-like. Doesn't seem to be a direct target, but it's some indirect target because we see it induced at later time points. But once it is induced, it's a very effective blocker of HES1, and thereby essentially shortcutting the whole system and shortcutting the notch pathway, literally making these cells insensitive to any upstream activation of the pathway and allowing these cells to further differentiate. So we wanted to sort of verify this or take a look, but this is actually happening during normal development. And Moritz did some in utero electroporation assays where he electroporated hairpin constructs against MYD1-like to knock down the gene. And as you can see, just based on migration, so down here is the ventricular zone and up here the cortical plate, not many cells actually make it up to the cortical plate when they are transfected with these hairpin constructs, suggesting that there is a differentiation block, and this is corroborated by various immune stainings to demonstrate that indeed acute loss of MYD1-like or reduction of MYD1-like in these ventricular precursor cells lead to a deficiency in their differentiation. So it seems that's what's going on in vivo during normal development. All right. As you can tell, we are very excited about this transition, and we are shooting with various methods at this question. And we also did a single-cell RNA-seq approach by forming an RNA-seq analysis of the reprogramming cells, starting from undifferentiated or uninfected fibroblasts all the way to about 3-week-old mature AN cells. And this is the distribution of the cells in between. As you can tell, we have sort of drawn this curved line here, which seems to be the reprogramming path. Here are the fibroblasts, those are the intermediate cells, here the mature AN cells. And there's a lot of interesting things obviously happening, but what we in particularly notice is there's 2 offshoots of these cells, looks like, from the normal reprogramming path. So this offshoot here is interesting because these cells seem to not really be able to go far and still have a lot of fibroblast features in them, perhaps are sort of on the way back to fibroblasts. And it turns out that it's exactly those cells that for some reason turn off ASO1 very rapidly. So I didn't tell you yet that the colors here indicate the levels of ASO1. And at that stage, it's mostly driven by the transgenic, expression from our lentiviruses. Not so much contribution will stem from the endogenous ASO1. So for some reason, these triangles here indicate day 5 of reprogramming, as early as day 5 after infection. And those are really strong, DOX-inducible lentiviruses. There is no ASO1 mRNA detectable anymore in these cells. We think it's, well, no, we actually have no idea why this is silent. We know there's a lot going on the protein, but also, as you can see on the RNA level, and it's not, has nothing to do with the technical problem. So GFP is highly expressed or other genes. So it's really ASO1 specific, but remarkably on the RNA level. So what I was about to say earlier is we think that HES1 might play a role in there. So, but from a reprogramming point of view, it's potentially a trivial explanation of why these cells don't reprogram, and they are actually majority of the cells, and that's why we end up overall in only about 20% efficiency, and probably most of these 80% are explained by this phenomenon. But there's also an interesting second offshoot, which is a little smaller, but it's shown here, but it's going further down here, and as you can see, the levels of ASO1 are still very, very high. So clearly cannot be explained by this simple technical explanation. So when we look at these 4 or 5 cells here, we see that there is a lot of muscle genes being induced in these cells. And that reminded us that ASO1, after all, is very closely related to this famous transcription factor called MyoD, which was found in the late 80s to convert fibroblasts to neurons. So coming back to this old papers, we realized actually we used the exact same cells that MyoD was discovered in. And we have now this really beautiful system that we can start with the exact same population, put MyoD in these cells and turns them into one way, that turns them very efficiently into muscle cells. Or we put ASO1, sequence-related BHLH factor, and we go the exact opposite direction and turn these cells into neurons. So we thought perhaps we can learn something when we compare these 2 transcription factors a little better. So again, we first made sure that the system is working. So we infected fibroblasts with ASO1 or MyoD, stained for neuronal and muscle markers, and as you can see, system works as advertised. ASO1 makes neurons, MyoD makes muscle cells. When we look at where ASO1 and MyoD is bound in these fibroblasts, we came to the remarkable conclusion that the binding is again very, very similar between these 2 factors. When you look at the motif that is preferentially enriched at these binding sites, we find the exact same type of epochs, namely the GC type epochs enriched in these sites. So it looks like biochemically, or sort of sequence preference-wise, these two factors are actually very, very closely related. And how can you explain these two different reprogramming outcomes by having a seemingly identical binding pattern in fibroblasts? We just confirm that everything is working well, and when you look at the RNA, by RNA-seq, now we see, of course, dramatic differences, as you would expect. One induces a muscle program, the other factor induces a neuronal program. So we have this discrepancy between binding and transcriptional response. And took us, again, quite a long time to look at sites and sort them by various parameters and see whether we can make any sense out of this. And finally, we came to the conclusion, actually, binding is important after all, but you have to sort of include a bit more quantitation and not only look qualitatively whether a factor is there or not, but also how strong the binding event is. And when we do that and incorporate the affinity, so to say, of the transcription factors into the equation, now all of a sudden things make a lot of sense. So those here are all the binding sites that are co-bound by ACE1 and MyoD, but sorted based on intensity, essentially. So up here, we have the sites that are preferentially bound by ACE1 and to a lesser degree by MyoD. In the middle, the ones which are co-bound similarly, and down here, the MyoD-enriched sites. And now, when we look at the changes on the chromatin, for example, when we look at an enhancer mark, H3K27 acetyl mark, and now these ACE1 preferred site, preferably bound sites, they open up. They are getting acetylated when you express ACE1 and not when we express MyoD. And the opposite is true for MyoD. So the MyoD-enriched sites are the ones that are actually responding, so to say, to MyoD, but not to ACE1. And we see a very similar pattern when we use ATAC-seq here, and even to some degree also the expression. But it's, of course, the target genes are hard to know for sure because the binding events oftentimes happen in very distal elements, so it's not always easy to identify the exact target genes that are regulated. But even on the RNA level, we see enrichment of these sites. So that's sort of satisfying that after all a biochemical event can explain something. But it also allowed us to dig a little deeper and compare these different sites. So this slide may look boring, but to me at least, it's the most exciting slide of my entire talk. So please bear with me to go through these data. Oh, sorry, this actually is kind of boring. The next slide looks similar though. Because this is essentially a quantification of what I just showed you, the average plots here of the heat maps, or the heat maps I just showed you, and the average plots here now that the it was ACE1 enriched sites are opening up, and the in ACE1 reprogramming, and the MyoD enriched sites specifically open up in MyoD reprogramming. This is the slide I was talking about. Looks even more boring, doesn't it? So up here we have the ACE1 enriched sites. Here the co-bound, and here the MyoD enriched sites. In this column here, it shows you what type of motifs we find under these sites. And this here shows you the significance, and this here shows you the occupants. How many of these sites actually have that given motif? Okay. So when we now focus on the ACE1 sites, we pretty much only find BHLH E-boxes, right? And ACE1 type motifs there. And the significance is very, very high. And it's a lot, in some cases, over 90% of the sites that are ACE1 enriched that carry these sites. When you now look at the MyoD enriched sites, the situation is very different. First of all, the significance drops quite substantially. And now, in addition to E-box motifs, we find other types of motifs, in particular a homobox motif, co-enriched to somewhat similarly significant levels. And when we look about the distribution or the fraction of the sites, it's actually not that many sites that even have an E-box. under these MyoD preferred sites, and to some degree even a greater enrichment of these homeobox motifs. Before I sort of try to explain this to you, let me just continue here. We also looked at all possible e-boxes. So the two base pairs in the middle are interchangeable. So that's why you have 16 possible e-boxes. And this here is the distribution of the three groups of target sites, MyoD, middle, or common, or AS1 enriched. And as you can see, it's the exact same e-box, the CG type e-box, that is the best binding site for both MyoD and AS1. But when you look at the frequency, AS1 is much, much more present than MyoD. So we took then this best binder, so to say, the GC type e-box, and plotted the number of these type of e-boxes in these three different groups. And as you can see, there is actually oftentimes many more than one e-boxes at the AS1 preferred sites, but taken together, at least one in almost all of them. But when you look at MyoD, about half of the sites don't even have the best affinity binding site, and there's, what is it, like 30 or so % that have only one, and very few more than one. And that cannot be just explained because on average the groups have different lengths, the peaks have different sizes, because the AS1 peaks actually tend to be somewhat smaller. Right? It cannot be statistically explained to have more of these motifs just because of the length of the peaks. So the reason why I got so excited about this is because this suggests a potential mechanism how a pioneer factor can find its proper targets. I told you earlier that ACR1 is this amazing on-target pioneer factor, sort of can seemingly ignore the chromatin configuration and just goes in, activates its neuron program, right? So for the longest time, I and many other people, I think in the field, have thought, well, these pioneer factors must have some special domain, right? There must be some sort of pioneer factor property built into the sequence, hopefully that we at some point can identify and put onto other transcription factors and make them pioneer factors. And these data suggest otherwise. These data suggest that pioneer factors are actually kind of stupid. In a sense, all ACR1 is able to do is finding the e-box. It can find the e-box really, really well, much better than MyoD, for example. But that's all it can do. It doesn't seem to be able to confused or affected much by other protein-protein interactions. May not listen much to where other transcription factors are going. You have to remind yourself that there's many, many transcription factors being present at a given time point in any cell. So this chromatin, these enhancers, they're full of a lot of different transcription factors. And you could easily imagine that in this enhancer milieu, it's not only the transcription factor, the protein-DNA interaction that is maybe most dominant in most cases. There's a lot of possibilities for the transcription factors to interact on the protein-protein level. And as I just showed you, half of the MyoD preferred targets where MyoD is physically present, there's not even e-box there. It cannot bind the DNA at these places. It must be, these interactions, or this binding, must be primarily driven by protein-protein interactions with presumably, to some degree, the chromatin, I guess, in the tighter sense, but in the wider sense, with other sequence-specific transcription factors, such as perhaps an homeobox transcription factor that is meant to actually bind the DNA there and can sort of recruit MyoD to these sites. So in theory, you can turn any ordinary transcription factor into such an amazing on-target pioneer factor by just wiping out or deleting all the protein-protein interactions that would be there for interacting with other sequence-specific transcription factors, and maybe to some degree also the chromatin. So it could be a very simple explanation for a very complicated process. Did everybody get that? No? Good. Yes, we have some further evidence, but those are hard experiments, or fun experiments, but sort of hard to make clear conclusions. But we also played around with the domains between MyoD and ASA1. And turns out that MyoD is, of course, a very well-studied protein, that there is actually a homobox factor known, PBX1, that is known to be important for MyoD function. And its interaction domains has been mapped to 3, I believe, amino acids, and 2 of them are in the C-terminal domain. And we found that we can actually turn MyoD into a pro-neuronal factor by just exchanging the basic domain, which is the DNA binding domain. So protein DNA affinity is important after all. But also when we then also co-exchange the C-terminus, which is just, I think, 30 amino acids or so of ASA1 with MyoD. So that at least is compatible with this idea that I just tried to bring closer to your attention, that we essentially can convert MyoD into a pro-neuronal factor by primarily by doing 2 things: increasing the affinity to its proper DNA binding domain, but also by eliminating or at least changing the protein-protein interaction domain with other sequence-specific transcription factors. And not surprisingly, it fits that this chimera construct now has a much more similar binding to ASA1 than to MyoD. All right, so this we found quite interesting, but it had one more potential consequence, which is I showed you that ASA1 is this very strong neuronal inducer, but it also has the ability to low levels at least to also induce a muscle program, right, as I just showed you, if it's there alone. In the normal reprogramming, we don't have to worry about it because we co-express MYD1-like, which takes care of the muscle program. So that's why we have this beautifully non-confused neurons that we reprogram with the 2 factors. But then we thought perhaps something similar can happen with MyoD as well. It's a well-known strong inducer of the muscle lineage, but perhaps, given all its similarities, perhaps it can also somewhat inefficiently induce a neuronal program. And perhaps you can sort of change the balance of these 2 sort of sets of target genes by taking MyoD and now co-express MYD1-like. Because MYD1-like, as I told you, very clearly doesn't touch the neuronal program, but it very efficiently blocks the muscle program. So what would happen if we co-express those 2 factors? Can we then turn MyoD into a pro-neuronal factor? And the answer is, as you can guess, because I showed you already the schematic, it worked. So those are not the most beautiful neurons I've ever made, completely admitted, but we were really stunned to see that you actually get some neurons. So just as a reference here, on the left, you see ASA1 alone or ASA1 with MYD1-like, and we get this really amazing, complex-looking neurons with high efficiencies. MyoD alone rarely induces like a Th1 positive cell, but with MYD1-like now, first of all, we seem to completely eliminate the induction of desmin and other muscle markers. So it's very, very few muscle cells being made. And now we see this kind of cool-looking, not quite mature, but still clearly neuronal-looking cells made from MyoD. And when we patch from these cells, they even have a fairly narrow action potential. It's too few cells, and not great cells. They won't have synapses, so you can't assess that function. But it's intriguing to see that we can make this type of cells with MyoD and MYD1-like together. All right. I'm at the end. This slide always reminds me that I'm so happy to be a scientist and leading a group of amazing people. We are having fun not only with our science, but also privately in the lab, as you can see. I just would also mention some names. I think I mentioned Moritz already, who was really in charge of the MYD1-like project, but he also teamed up with a GNE, a student in the lab, who worked on the MyoD ASA1 comparison. And Jean and Koji did this blood iron project. A lot of the ATAC-seq stuff was done in collaboration with Howard Cheng. Thank you very much for your attention, and if you have any questions, I'm happy to take them.
Thanks so much. Great talk.
Thank you.
So following on this DNA binding factor specificity, so We know that FOXA1, for example, was a classical one, and it found to bind the entire motif. And then Kenzarites and others show that OCT4 and SOX2, for example, they bind the partial nucleosome motif, which actually much broader than their final sort of set of binding sites. So maybe I missed it, but what with an ASCL1? Does it actually bind? You show the TN5 transposition open chromatin, but so does that actually ASCL5 binds its original set of sites and then sits there, or it's actually much broader and then it's condensed to what it should be?
Thank you for that question. We are amazed by these data. So Kenzarite and also Catherine Platt up in a little further north of Southern California, in Los Angeles, I guess, showed very elegantly that OCT4 is also a pioneer factor based on the definition that it can access closed chromatin. When you though look and compare the binding sites 2 days after infections in fibroblasts and where OCT4 is bound in ES cells, it's a very different binding pattern. So it's more like our brain2 factor, very much context dependent. So it can access closed chromatin, but it's, if you want to say, the wrong sites. And Catherine's paper suggests that it's actually mostly fibroblast enhancers where OCT4 is going, and she thinks that might be a reason why the fibroblast program is shut down. So OCT4 is responsible to actually shut down the fibroblast program first, and then at the later phase of reprogramming, then induces the pluripotency program. So the binding is very different of OCT4 in the early and late phases of, and that's completely different for our ASA1. It goes right in and stays there and activates the neural program. And I actually asked Ken, after he published this paper with this half, which is mechanistically very interesting, right? It looks like maybe there's just 3 of the 6 base pairs are accessible for a pioneer factor, so that tries to grab on. But if it's the wrong motif, what do you need the pioneer factor there for, right? I mean, I guess it has some function, but it's not the proper target program, right? So there seems to be a fundamental difference between most of the described pioneer factors and ASA1.
So have neuronal genes evolved to have multiple E-boxes that ASCL1 can bind to of the GC type?
Yes, I guess in principle, BHLH factors can bind all 16 possible E-boxes, but as I showed you, there is a clear preference of this CG type E-box for ASA1 and MyoD.
And do you know anything about the spacing of those E-boxes? Is there some sort of DNA structure or oligomerization of ASCL1 that, for instance, from the C-terminus or wherever, that drives it?
Yeah, we haven't looked at the distribution of the E-boxes. I told you that there's oftentimes more than one, actually in most cases for ASA1, actually more than one E-box is there. So it seems to be a cluster of E-boxes, which could maybe explain, why there's so many E-boxes in the genome. Why doesn't ASA1 bind everywhere? It may go, those sites have like maybe 3, right, or 4 E-boxes, and that's why if ASA1 is primarily driven by the DNA interactions and less by the protein interactions, that's maybe the highest affinity site where it goes. But that's just a hypothesis.
Well, affinity is always helped by multiple sites, right?
Yeah, sure.
My question is actually similar in nature in that, Alex Meiser for FOXA, which has also been described as a pioneer, he has shown that when you infect fibroblasts, I believe he used too, with FOXA, that there's sort of these low affinity sites and actually scans sort of all its possible binding motifs. And then, some evolve to be sort of these more high affinity sites doing reprogramming over time. And the idea is then that that's sort of facilitated by cooperative binding. And you describe here that ALC1 essentially all goes to these neuronal sites. When you sort of lower your threshold, do you actually also see that maybe it goes to non-neuronal other sites? And then is that maybe that it sort of binding becomes enhanced because it maybe homodimerizes, and it's almost cooperativity with itself, and that's why it's reinforced at these neuronal sites.
Yeah, interesting idea. First of all, I would like to make clear that I was a little bit painting black and white, right? Just to make the point a little clearer. But of course, it will not be that extreme. And of course, it will be somewhat context dependent and dependent on other sequence-specific transcription factors and so forth. Just to make it a little clearer, right? I was trying to make sure. Biology is always gray, never black and white. So first of all, we know that ACE1 is usually not homodimerizing. So it takes a constitutively expressed E protein to bind. So that's why some sort of feed-forward mechanism, like you described, I'm not sure we could explain that easily in this. I think it looks like the data more compatible with the idea that simply the highest affinity sites that ACE1 is going to. We also try to do some imaging now, single molecule tracking and so forth, to see, look at the dynamics. Maybe there's differences between these different types of transcription factors. Maybe we can learn something that way.
Beautiful talk. I'm just wondering, in principle, when cell change fate, establish a new identity, there are all kind of reorganization of the genome happens, right?
Sure.
And now the question is that this endogenous gene, all 3 of them or many others, how many things become dispensable after initial reprogramming? So in the other word, you don't need a consistent expression of a foreign exogenous gene and then replaced by the internal program of endogenous gene. So when this has happened, which one is persistently required or none of them?
Yeah, so we have looked at that question, and as you said, as you would expect from a proper reprogrammed cell, these ion cells are not dependent on the exogenous expression of the viruses anymore, or the transgenes, whatever method you use to.
What window? 1 month, 1 week, 1 month?
So we did a titration, and essentially the shorter you give doxycycline, the fewer cells you make. I think we could make some neurons with as little as maybe 3 or so days of doxycycline. Was very inefficient. So it depends, what efficiency you want. Then I can give you an exact time of how long the transcription factor are needed. But it's very clear that the neurons that are made, independent of the efficiency, they are not dependent on the transgenes. And in case of iPS cell reprogramming, the solution is very easy because the reprogramming factors, OCT4 and so forth, they are being induced. So the endogenous OCT4 takes simply over, and that's why you don't need OCT4 anymore, or SOX2 and so forth. That is somewhat different in our case because ACE1 during normal development is only transiently induced, is expressed right at the transition when the neuroprogenitor cells have the last cell division and become differentiated neurons, or these bipotent TBR2 positive cells. And then it's turned off. But there's a lot of other BHLH factors in the genome, and there are some, in particular some NeuroD family members that then are being induced. So it's probably a little bit more complicated in terms of neuronal formation than pluripotent cells, that there's more transcription factors on, it's a sort of different related program probably that is being induced. It's not the exact same identity of the transcription factors that we use to reprogram. But one leg, on the other hand, stays on. So it's endogenous transcript and protein is being induced and stays on, and takes over.
Okay, if you want more philosophy, come.
Yes, I'm happy to try to philosophize more later on.